To all audience, please be seated. We will begin the program shortly. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Good morning, ladies and gentlemen. Welcome back to Day 2, Postgraduate Collquium 2025, Translational Biomedical Research, bringing the gap from bench to bedside.
We hope that you enjoy yesterday's event, filled with inspiring presentation and engaging discussion, also ideas for future collaboration. And this event is made possible by a collaborative work between Postgraduate Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, and the Faculty of Health Sciences, University Kebangsaan Malaysia.
We would like to remind you to cast your vote for favorite poster by scanning the barcode you find on the signage outside this room. The voting will be closed at 10. You can also see the poster directly as they are displayed on the screen.
Ladies and gentlemen, allow us to briefly walk you through the agenda for today. It will be started with 2 plenary sessions featuring distinguished speakers from both universities, followed by Q&A sessions.
And the second, Rapid Fire Session 3, will begin right after the plenary sessions.
Then, we will have a tea break whilst our judges will gather to determine the winners from rapid fire and poster competition. You can use the lounge at the back of this room to enjoy your coffee and snacks or pay the last visit to the poster display.
And our day will be concluded by a closing ceremony in which awards for the winner from each category will be announced. And now, let us begin with the first agenda. Plenary Session 3.
This session will be moderated by Professor Dr. Rerum Naturalium Asmarina, MSc. We are going to briefly read her curriculum vitae.
Professor Asmarina is currently a senior professor at the Department of Medical Biology, Faculty of Medicine, Universitas Indonesia, and serves as the head of the doctoral program in biomedical sciences. Since 2024, she has also been appointed as the chair of the Indonesian Consortium in Biomedical Science, or KIBI.
Her academic journey began at Universitas Indonesia, where she earned her bachelor's degree in biological sciences in 1989. She continued with a master's degree in biomedical sciences in 1996, and then completed her doctoral degree for Dr. Rerum Naturalium in 2003 from Justus Liebig University of Gießen, Germany, specializing in molecular and reproductive biology.
Professor Asmarina has more than 33 years of academic and professional experience. She has been a lecturer since 1990, became a senior lecturer in 2003, and has held numerous leadership positions within the faculty, including research coordinator and head of the molecular biology laboratory since 2013.
In terms of research, Professor Asmarina is an exceptionally productive scientist with over 90 international publications and more than 40 research grants throughout her career, both as principal and co-investigator. Her work focuses on genetics, epigenetics, reproductive biology, and molecular medicine.
She has received multiple honors, including the Satya Lancana Karya Satya, 10, 20, and 30 years awards from the President of the Republic of Indonesia, and international scholarships such as the AAD Fellowship from Germany.
Additionally, she holds 3 intellectual property rights and has contributed to several scientific books and educational modules. Everyone, please welcome Professor Asmarina to the stage.
Thank you very much for the introduction. So, Bismillahirrahmanirrahim. Good morning, very good morning for all the participants. Yeah, how are you today? Fine, especially from our guest from Malaysia. It's fine. Okay. Where was you in the last night? Monash. Okay, it's nice place in our city. Yeah. Okay. Yeah. Today we have 2 distinguished speakers. Yeah, from our university, from Universitas Indonesia, and also from UKM. So for the first speaker, yeah, please we invite Professor Dr. Novi. Please the CV. And then Professor Novi will present the topic about the cellular senescence in obesity. How can it be prevented? So here is the curriculum vitae from Professor Dr. Novi Sylvia Hardiani M. Biomed. Yeah, should I read the CV first? So Novi, yeah, we call as Novi, Professor Novi. So she is a medical doctor, graduate from medical doctor from our faculty, and then 2007, yeah, she graduated for a master's degree in biomedical science in our faculty also, and 2012, yeah, she also graduated from our faculty. Now she is head of division of research and collaboration from the National Organization for Biomolecular, yeah, Biomedical. So I don't know how many publications, but I see in this, her CV, there are about 60 publications, yeah, from Professor Novi. So yeah, we give you time, about 20 minutes, Professor Novi. Thank you very much.
Okay, thank you, moderator, for the introductions. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, I'd like to extend my gratitude to the organizing committee for these invitations to speak today. It is truly an honor for me. Yeah? In this presentation, I will explain about cellular senescence in the context of obesity, along with the preventive strategy based on insights from the literature and also finding from our research. Okay, for the introduction, obesity is characterized by excessive accumulation of adipose tissue, and it is a major contributing factors in the pathogenesis of degenerative disease. And the prevalence of obesity continues to increase. Yeah? More than 1 billion people now living with obesity, and about 2.8 million people die each year with obesity. Therefore, the obesity considered as the epidemic in the world now. And in Indonesia, yeah, over the past few decades, the prevalence of obesity saw to sharp increase across all age groups. Yeah, according to the Riskesdas data, the prevalence of obesity sharp rise in adult, yeah, from 28.9% in 2013 to 35.4% in 2018. Yeah. Okay. Obesity condition is associated with the low-grade chronic systemic inflammation in adipose tissue, originating from the excessive adipose tissue. Adipocytes secrete pro-inflammatory cytokines such as tumor necrosis factors alpha, And these cytokines stimulate the production of ROS, such as anion superoxide, hydrogen peroxide, and hydroxyl radicals. And the accumulation of ROS lead to oxidative stress, damaging our macromolecule, such as lipid, DNA, and also carbohydrate. So it is really dangerous for our cells. And the oxidative stress and the inflammation process promote cellular senescence in obesity. And cellular senescence is a key factor in the pathogenesis of degenerative disease, yeah, especially type 2 diabetes mellitus. Yeah. Slide. Okay. Cellular senescence is a condition in which cells stop to divide and cannot longer return to their original state. Yeah? And the cell cycle itself is regulated by protein kinase, which is known as cyclin-dependent kinase, CDK, which play a role in activating retinoblastoma protein, or PRB. And this PRB is responsible for the progression of each phase of its cycles in cycle cells. Yeah? And all somatic cells with proliferative potentials are susceptible to undergoing senescence. Senescence cell is stimulated by various exogens, yeah, such as DNA damage, telomere shortenings, oncogenic signals, ROS accumulation, and inflammation. Yeah. And the senescence process began with the activation of P16 and P21. It is a protein which inhibits CDK. The inhibition of CDK lead to inactivation of PRB, resulting cell cycle arrest. Yeah. In addition to cell cycle arrest, senescent cell also secrete the SASP, senescent-associated secretory phenotype, which consists of cytokine, chemokine, MMP, and also growth factors. This SASP exert harmful effect to the neighboring cells because it can stimulate senescence in the neighboring cell and also can stimulate tumorigenesis. Yeah? And also based on its cause, senescence cells divided into two types. The first type is replicative senescence, and the second type is premature senescence. In replicative senescence, disease occurs due to cellular exhaustion because of repeated replication cycles. It is caused by progressive telomere shortening because of repeated cell cycles. And in other hand, in premature senescence, the cells senescence occurs due to the exogenous stressors such as oxidative stress, which is happen in obesity. Slide, please. Okay, a set of general hallmarks of cellular senescence has been defined as seen in these pictures. The first one is stable cell cycle arrest, yeah, which is originating for the inactivation of PRB, yeah, because of P16 and also P21 activation. And also there is the chromatin alteration and reorganization. And for the morphology, there is the enlarged and flattened morphology, and followed by macromolecular damage and metabolic changes because of ROS accumulation, and increased lysosomal compartments. This is signed by overexpression of S-abigal galactosidase is the enzyme in lysosome, which is overexpressed in cellular senescence. And also senescent cell secretory associated senescent phenotypes, SASP, yeah, that I already explained before. Yeah. There's so many biomarkers, yeah, to detect cellular senescence using various methods such as immunofluorescence, immunohistochemistry, or Western blot. Yeah. We can detect cell cycle arrest by measuring expression of P16 and also P21 by immunohistochemistry or ELISA. Yeah. The result is the increase of this expression so that the cell going into senescence. And also, we can also observe the structural morphology. There is the enlarged and flattened morphology. and also increase the lysosomal compartments, which is characterized by the increase of S. abigails, yeah, and then adimase, and also we can check the ROS accumulation or oxidative stress markers. And the other parameter is telomere shortenings, yeah, and also SASP cytokine, yeah, by ELISA method, and also the others is lamin B, and we can see in these tables here. Okay, in obesity, cellular senescence is the multi-step process, yeah. Early senescence can be initiated by the overweight condition, yeah. Overweight is caused by sedentary life, yeah, such as Western diet, and also lack of exercise, yeah, also the genetic factors, yeah. In the early of senescence, there is a changes in the chromatin, in the cell, yeah, there is the remodeling, and also the cell secrete SASP cytokine, yeah, and the lysosomal compartment also increase, signed by S. abigail characterization, yeah. If the overweight condition cannot overcome, it will continue to the obese, yeah. In the obese, this is the chronic low-grade inflammation, yeah. So the stressor persistence to the cell, and the cell will enter late senescence. All these conditions responsible for the occurrence many kind disease, yeah, including in the brain, in the liver, in the pancreas, and also in the intestine. Slide, please. Okay, our research proved the cellular senescence in obesity, that senescent cell accumulate in various tissue. We did the obese rat using high-fat diet for 24 weeks, yeah, by giving the about 48% of fat, yeah. And our research find that the cellular senescence is detected but in brain, in heart, peri-aortic adipose tissue, and also in the liver, yeah. In the brain, yeah, there is the increase of P16, the expression is significantly increased, and also SA abigail galactose increase. And this premature senescence is perhaps driven by the oxidative stress and inflammation, as evidenced by the increase of malondialdehyde. Malondialdehyde is the marker for oxidative damage for the lipid oxidation, and also followed by the increased interleukin-6 and the decrease of glutathione and also catalase. And our research also proved that the cellular senescence in the brain accompanied by the brain damage, which is signed by the increase of neuron-specific enolase, NSA. Yeah, the NSA is biomarker for neurodegeneratives, and it is detected in the Alzheimer and also Parkinson disease. Besides in the brain, cellular senescence also detected in the heart, yeah, which is signed by the increase of S. abigail and also the presence of lipofuscin. Lipofuscin is the hallmark of cellular senescence, yeah, which is from the oxidized compartments, yeah, which cannot degraded by lysosome, yeah. This is the brownish granules, yeah. And also oxidative stress also increase in the heart, yeah, signed by the increase of MDI and also the decrease of antioxidant. And in the peri-aortic adipose tissue, also cellular senescence detected by the increase of P21, S. abigail, and also the lipofuscin, yeah. The senescence in adipose tissue can impair the adipocyte function, yeah, and also can impair the adipogenesis and enhance lipolysis. Therefore, free fatty acid will increase in the cell, and the increase of free fatty acid can disrupt the pancreas cell beta pancreas, leading to diabetes mellitus. Yeah. And also in the liver, we found the senescence as evident by the increase of P16 and S. abigail, and oxidative stress by increasing MDA and the decrease of catalase. And also, this cellular senescence in the liver lead to the liver dysfunction, as evidenced by the increase of alanine, ALT and AST, and also accompanied by non-alcoholic steatohepatosis, which is observed by histological examination. So, because the solar senescence in obesity is really harmful, yeah, so we have to need the strategy to prevent solar senescence and reduce senescence cell build-up, yeah. Of course, the first strategy is do healthy life modifications such as healthy diet and exercise, yeah, to reduce body weight, but usually this is challenging for the obese individuals, yeah. So the second strategy is the calorie restriction. One of the calorie restriction is intermittent fasting. And the other strategy is inhibition of oxidative stress, because the solar senescence is caused by oxidative stress, by antioxidant administration. And also the last strategy by using xenotherapeutics consists of senolytic to eliminate senescent cell, and also xenomorphic to inhibit SASP. Okay, calorie restriction is understood as a method of reducing calorie intake without causing malnutrition. And several studies prove that calorie restriction up to 20 until 40% can extend lifespan and create healthy aging, yeah. But the research still limited in the animal models, yeah. The molecular mechanism how to calorie restriction can create healthy aging by the activation of FOXO3. FOXO3 is a transcription factor which regulates many kind genes such as antioxidant enzyme and also cell survival, yeah, and proteostasis and autophagy. And the activation of FOXO3 associated with the prevention of degenerative disease. And also calorie restriction can activate sirtuin-1. Sirtuin-1 is the acetylase, yeah, which regulate the mitochondrial biogenesis. The activation of sirtuin-1 can activate PGC1 alpha. PGC1 alpha is coactivators which are responsible for mitochondrial biogenesis. So it can inhibit risk of age-related disease. So healthy aging can achieve. Next slide, previous slide, please. Okay. Intermittent fasting is a type of fasting which include eating patterns of little or no energy intake for an extended time period, generally lasts 16 hours until 48 hours, with intervening windows of normal food consumption on a recurring basis. Actually, there is several types of intermittent fasting, such as complete alternate day fasting and time restriction feeding, and also Ramadan fasting and other religious fasting. We can see here both calorie restriction and intermittent fasting is similar in stimulating molecular mechanism. It can inhibit mTOR and activate FOXO3 and also sirtuin-1. All these mechanisms can inhibit aging and can create healthy aging. Please go back to. Okay, we did the research using New Zealand white rabbits, yeah, to explore the role of intermittent fasting on the FOXO3 expression, IGF-1 expression, sirtuin-1, and SOD in the liver of New Zealand white rabbit, and also to check the oxidative stress parameters such as MDA, carbonyl, glutathione, and catalase in heart, brain, and kidney. 15 of New Zealand white rabbit is divided into 3 group: control and intermittent fasting group and prolonged fasting group. IF group was fasted for 16 hours, followed by non-fasting period about 8 hours. And the PF group was fasted about 40 hours, followed by non-fasting period 8 hours, for 6 days. And in the day 7, the animal was sacrificed and checked for these parameters. Okay, our research proved that intermittent fasting group can increase mRNA FOXO expression and also mRNA sirtuin-1 expression and FOXO3 protein expression, and also the increased SOD, yeah, compared to the control and prolonged fasting group. And also IGF-1 is decreased, yeah, both plasma and hepatic IGF-1 in intermittent fasting group. And we speculate that intermittent fasting is a low energy status. It will increase NED, and this condition will activate sirtuin-1, and activation of sirtuin-1 will deacetylate the FOXO3 expression. In addition, FOXO3 expression also stimulated by the decreasing of IGF-1 expression. Because of FOXO3 is a transcription factor for antioxidant expression, so SOD activity increase here, and oxidative stress will be inhibited. Okay, and in oxidative stress parameters, in heart tissue, we found that MDA was significantly greater in the control than in the IF and PF group. And in brain tissue, glutathione was significantly higher in IF group than in the PF and control group. Also, catalase was significantly higher in IF and PF than in the control group. However, in the kidney, catalase specific activity was significantly lower in the PF than in the control group. We conclude that fasting reduces oxidative stress, and IF or intermittent fasting appears to be more effective than prolonged fasting in protecting this vital organ from oxidative damage. Besides animal model, we also did randomized control trials conducted in 50 obese males aged 19 until 59 years old, yeah, divided into two groups. Control consists of 25 subjects, and intermittent fasting group. In this group, the subject do the fasting 2 days. It means that the fasting do in Monday and Tuesday for 8 weeks, yeah. And then both of group give the education for balanced diet, yeah. And prior to and following the intervention, whole blood sample were collected, yeah, to analyze the MDA and FOXO expression, proteasome activity, and others marker for oxidative stress such as carbonyl, glutathione, and total antioxidant capacity. We can see here that the intermittent fasting group, yeah, have the lower body weight, yeah, significant lower body weight compared to the control. However, there is no change in BMI, body mass index, and also the MDA level is significantly lower in intermittent fasting group after doing fasting for 8 weeks, yeah. And also we check the FOXO3 expression. There is the increase of FOXO3 expression and also proteasome activity. Proteasome is the protein complex which maintain protein homeostasis by degrading aggregate protein, yeah, to prevent aggregation. And also the decreased carbonyl, the increased glutathione, and total antioxidant capacity. Okay, the second strategy is to inhibit oxidative stress by antioxidant administration. Yeah. Antioxidant can be found in the herbal, which is contain polyphenolic flavonoid compound, such as coriander, yeah. Coriander sativum have the ability as the antioxidant and anti-inflammation. So this is potency to remedy for obesity. We did the characterization of coriander seed. We detected flavonoid components of coriander seed using LCMS. There is alloresin, camphor, quercetin, and viscumnoside. Yeah, and also using GCMS, there is several monounsaturated fatty acid, yeah, such as succinic acid. In order to elaborate the role of corianders in eliminating oxidative stress, we administered the 100 milligram per kilogram body weight of coriander seeds extract to obese rats, yeah, induced by high fat diets for 12 weeks, yeah. And there is the 5 group. The first group is control. The second group is the normal rat with coriander seed. And the third group is normal rat given with high-fat diet and coriander seed. It is the preventive group. And the fourth group is high-fat diet group. And the last group is obese rat with given coriander seed. We found that in the fifth group, which is the obese rat with given coriander seeds, the MDR and interleukin-6 level decrease, followed by the increase of specific activity and catalase enzyme, and also glutathione in brain. Previous slide, please. Previous slide. Go back to slide, please. Go back to previous slide. Yeah, and then the administration of 100 milligram per kilogram body weight of coriander seed ethanolic extract in obese rat also decreased cellular senescence, as evident by the decrease of P16 and S-abega levels in brain, as we can see here, and also the effect of coriander seed can be seen in the heart, and MDR level and also specific MDR level decrease, followed by the increase of catalase enzyme and glutathione, and also the cellular senescence is inhibited by the administration of coriander seed, as evidenced by the decrease of P21 in the heart, as we can see in these pictures. Okay, the last strategy is xenotherapeutic. Xenotherapeutics means that therapeutic utilize small molecule that specifically target senescent cells. It is divided into two, senolytic and xenomorphic. Senolytics selectively kill senescent cells. This is promote apoptosis, because senescent cells resistant to apoptosis caused by the pro-survival pathway activation. And the first senolytics, which already reported now, is combination of that dasatinib and quercetin. Dasatinib is the anti-cancers. It is a tyrosine kinase inhibitor, which can inhibit proliferation and also stimulate the apoptosis. Quercetin is the flavonoid component. It can stimulate apoptosis by inhibit anti-apoptosis pathways such as BCL-XL. And the second one is xenomorphic. Xenomorphic is inhibit SASP, because the SASP can influence neighboring cell to the senescent. So by suppressing SASP expression, using targeting NF-kappa B, mTOR, and the others cytokine, it can eliminate the senescent. Our in silico study tries to elaborate the potency of coriander seed in xenomorphic and senolytic. In the in silico study, found that the compound of camperol has the potential to act as an interleukin-6 inhibitor. Interleukin-6 is a cytokine, which is the main part of SASP component. So this is the effect of coriander seed in inhibiting xenomorphic by in silico study. Go back for the previous slide. Besides the xenomorphic, we also check the senolytic ability, and our in silico study evaluated the inhibitory potential of coriander phytoconstituent against several antiopotic protein, including BCL-XL, BCL2, BCL-W, and survivin. This is using molecular docking simulation. And we found that flavonoid component in coriander seed can inhibit the anti-apoptotic BCL-XL pathway involved in senescent cell anti-apoptotic pathway, or SAP. In order to prove in silico finding, we did the in vivo study. We give the 100 milligram per kilogram body weight of coriander seed ethanolic extract to obese rat induced by high-fat diet for 12 weeks. And we found that the cellular senescence is reduced in peri-aortic adipose tissue, yeah, as evidenced by the decrease of SAB Gals, and also followed by the increase, the decreased BCL-XL. This is anti-apoptotic pathway. However, it's not statistically significant, and also followed by the increased apoptosis in peri-aortic adipose tissue. However, it is not statistically significant. Yeah. And for the xenomorphic ability, fortunately, no significant difference, yeah, in SASP component, interleukin 6, tumor necrosis alpha, interleukin 1, and interleukin 10. Yeah, there is no significant difference between control. Yeah, therefore we conclude that in this dose, coriander seeds has no capability as xenomorphic agent. Yeah. So the senolytic and xenomorphic potential of coriander seed could not be confirmed in our study, potentially due to suboptimal dose and treatment duration. We only give 12 weeks, and the relative low level of flavonoid, particularly quercetins, and also the ability may have contributed to the lack of observable effect. And there is the natural product with higher concentration of quercetins, yeah, and camperols. It's the member of Allium genus. And in the literature stated that the outer skin of red onion or Allium cepa contain about 5,110 milligram per kilogram of quercetin and 481 milligram per kilogram of camperol. And also in scallions, there consists of 1,497 milligram per kilogram quercetin and 832 camperols. Given the high concentration, high content of these bioactive compounds, maybe Allium species represent promising candidates for senolytic and xenomorphic application, although this requires further experimental validation. Okay, conclusion. Okay, for the conclusions, aging is an inevitable biological process that progresses with increasing chronological age. However, cellular senescence can be accelerated under pathological conditions such as obesity. In obesity, senescence accumulate across multiple tissues accompanied by elevated oxidative stress, which contributes to the onset and progression of metabolic dysfunction and also degenerative diseases. Although lifestyle interventions such as calorie reduction and also physical activity is the foundational strategies for mitigating the effect of obesity. However, the long-term adherence really challenging for the obese individual. Alternative approaches such as the use of antioxidant compounds and the development of xenotherapeutics derived from natural products such as corianders, promising adjunctive strategy to prevent oxidative stress and cellular senescence, support the concept metabolically healthy obesity. Further study are needed to optimize the dose, improve bioavailability, and validate its efficacy to in vivo and clinical investigation. Okay, in the end of this presentation, let me introduce the coriander seed research team. Yeah, consists of the staff member from Department Biochemistry and also from Department Medical Chemistry, and also there is several PhD student and master student and undergraduate student. And also we collaborate with the Associate Professor Bimo Attejo from UPM. Thank you for your kind attention. Assalamualaikum warahmatullahi wabarakatuh.
Yeah, Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for interesting presentation with the so many report. We will discuss in this topic later after the second presenter. So I would like to summarize the presentation of the Professor Novi. So cellular senescence can give impact to, especially in obesity, give impact to our health, such as type 2 diabetes. Can lead to the type 2 diabetes, also cardiovascular diseases. So this cellular senescence obesity can be prevented by at least four strategy: calorie restriction, that also correlated with intermittent fasting, of course. Intermittent fasting it's mean that's calorie restriction actually. So and then antioxidant therapy, and then this last one is the xenotherapeutic strategy. So there are two mechanisms to impact for this xenotherapy are senolytic and xenomorphic. It is interesting that the natural product can also inhibit the senescence, cellular senescence, such as coriander. We use the coriander as spices, in our daily food, and also allium cepa. We use also many allium cepa in our food, So this is an interesting topic for Novi presentation. Now, please, the question later, okay, now the second presenter is Dr. Mazlizam, so from the UKM. The title of his presentation, Hastening Natural Products for Wound Healing, from Research to Clinical Application. So I think this topic also correlated with the cellular senescence, because the cellular senescence is also one of the mechanisms for wound healing. So now we continue with another topic, but it's correlated with the Prof. Novi presentation. So before I present, I would like to read your curriculum vitae. So Dr. Mazlizam bin Abdul Latif, the lecturer, staff from Faculty of Health Science from University of Kebangsaan Malaysia. So 1999, he graduated from biochemistry major from University Malaya in Malaysia for the bachelor degree. And 2008, he graduated from University Kebangsaan Malaysia, with the Doctor of Philosophy. So the work experience from him is the Biomedical Sciences Program Center for Toxicology and Health Risk Studies, Faculty of Health Science, University Kebangsaan Malaysia. And please hear the recent publication, 2024. She's published about the chromolina chordorata layer, nitrile rubber polymer transdermal patch enhanced wound healing. So 2023 also he published another publication with the infectious antibiotic methicillin resistant in Staphylococcus aureus. So I think it is also interesting topic, because wound healing can be also treated by natural products. So please, Dr. Mazlizam, time is yours for 20 minutes. Thank you very much.
Tingginya. Okay. Thank you to distinguished professors from UE, and UKM, also colleagues, friends, doctors, especially our students, postgraduate students. Thank you, and Assalamualaikum and Salam Sejahtera. Do I have the... So, my name is Dr. Mazlizam. The picture is before, for the introduction now is right. Okay, not on the poster, Okay, that's a very long time ago. Okay, so today I'm going to present, I'm going to speak about our work in natural products, okay, especially for wound healing purposes. Okay, and Uh, I come from Faculty of Health Sciences, UKM, and from the Center of Core. Okay. Okay. So as we know, there's a lot of challenge, especially in treating wounds, especially wounds which are chronics. It's very difficult to be healed. It is usually caused by infections, okay, and biofilm, which can cause the wound to resist treatment, and also probably then because of the very hard to heal, then there will be overuse of antibiotics, then will lead to resistance also for the healing of the wound. And then also the current standard treatment using drug or dressings, okay, have no defined way on how to treat certain wounds, okay. So lack of personalized treatment for different wound types, and also frequent dressing changes also can lead can disturb the healing process itself. Thus, because of this, as we know, it will increase costs, okay? Burdens on economics, especially, like in US also about 20 billion is annually for the treatment. In Malaysia also, is about the treatment cost per person for the chronic ulcer is about 6,000 per person. In private setting is about 10,000 ringgit per person. Per person. So in 1 year, there's going a lot of patients, so it's going to be millions in terms of spending, especially in the, what we call it, government setting, government clinics, okay? And to address that, we need an innovation, okay, promising alternative. So in this case, why not we look into the natural products, which have been used traditionally in our society long time before, okay? But with a little twist, probably, okay, we can incorporate it as a smart dressing, okay? And with the combination of, what we call it, antimicrobial setting by this natural product, probably they can give more protection for the wound to heal. So here, as we can see, okay, there's a lot of natural product that have been used, okay? So, okay, so it have many action, not only in wound healing, antibacterial, also it's a lot of, we call it, action that can be performed by these natural products. So now we're looking into the natural product. Okay, what we do in the lab. Okay, I'm going to represent this. Okay, why it is very important, because they can act as the in such a way that they can provide multiple mechanisms. Not only they can cross wound, they can provide antibacterial, antimicrobial, anti-inflammatory. So this combination can give advantage to help in the process of wound healing. And also because this is a natural product, okay, most of them generally well tolerated by human body, okay, and also because they come in the form of a natural product, they usually have synergistic effect, okay, cascading effect, which can help also the wound, okay, and it is what we call as the holistic, sustainable, and effective approach for wound healing, okay, with probably low cost, okay. So today I'm going to share our research on three promising plants and also one key compound that we have done in the lab. Okay, first is the Alocasia lanulata angler. Here is part of yam. It's traditionally used by indigenous people in treatment of wound, cutting wound. And then we have Illusion indica, or we call it as rumput sambal. This is the pest plant. I think you can find it anywhere. anywhere in Asia. Okay, then this is the zingiber zurumbate. Okay, one type of ginger, and also the active compound from the zingiber zurumbate, which called as zerumbone. So here, alocasia rectangular is a plant yam type, okay, and can be found in usually near the end of the jungle, okay, near the place where it is cold, okay, and traditional use is as anti-inflammatory topical application. Usually the native will, when they have a wound, they will take the stalk, batang, stem part, okay, and they will cut it and put it on the wound itself, and then they will tie it. So it will induce the process itself. It will, if they have a cut, it will jantung will kita mencantumkannya. Okay, and also okay, is what we have done on research of it, from the extract of it. It is not toxic, no IC50 value. We do the aqueous extract, okay, and we do the GCMS. They have a compound here which is have been proven from other plants, which also induce wound healing. And our lab finding also show wound healing potential. No IC50 here is a stalk that we use, the stem. Okay. So we have done the in vitro part. So when we do the in vivo part, we do it in the form of gel. Okay, for easy. That is the purpose of how to using it. Okay, for topical application. And it is found that it heals very good as par with control, which is commercially available, or probably better, much better. Okay, in terms of when we do the histological analysis, the collagen reproduction, the epidermal junction, the process of the reconstruction back of the skin is quite good, as comparable as if we are using the commercial available product that we use as control. And also when we do, this is the in vitro study, what we do the scratch assay. This is in vitro migration ways to detect whether that where this chemical have the ability to migrate the cell, to induce the migration of fibroblast cells, especially. Very important in our dermis to induce wound healing. So it induce, okay, it induce the fibroblasts to move, okay, to the wounded area in the plate. And also we do the analysis of the metal inside in the stem extract. We found it is high in potassium, which is very important, especially in the process of wound healing, potassium channel and everything. So also the study on the cell culture, okay, we do the G expression. It increases the expression of TGF beta, PDGF beta, BGF, and collagen type 3, which is very important in the reorganization of matrix of dermis, and also high potassium content, and we found out that it contains this, all this compound, which is very helpful, especially in inducing wound healing. So that is the story of alocasia. Okay. Okay. The next one is Eleusine indica, we call as the common plant, yet a powerful grass. It's a pest plant and invasive species. We call it as a rumput sambau. I don't know, in Indonesia it is called oh, okay. So this is a pest plant. If you have one in your lawn, it will be hard to remove, even you try to pull it out. So instead of throwing it, why not we do some research on it and find the its use, its importance. And as we know, in research before, this plant, it have anti-inflammatory, have been used traditionally also in a lot part of the world, from Africa until Asia. Okay, as anti-helmintic, anti-inflammatory, lot of anti-cancer. Okay. However, there is no definitive research reported on the activities of wound healing. That's why we very interested to go into it. So also it is used as traditional medicine in wound healing. What they do is they take the plant, the leaf part, and then they smash it, and then when they have wound or everything, they just paste it and bandage it and change it for to mitigate the process. And it is found it is rich in phenol and flavonoid, and it have antimicrobial and anti-inflammatory activity. So, but the earliest part of the research involved the GCMS analysis and also the toxicity test. So we extract it using the series of solvent, okay, from non-polar to semi-polar to polar, from ethyl acetate, ethanol and everything, and mostly it is found the compound extract is not toxic on the low dosage, okay? And also what we found is about 30 or more compounds which can be researched from the library that it exists in the extract itself, on all our extracts, okay, which can contribute in the wound healing process. So with that, we take the active compound and turn it into gel also, and to do the topical application and do the study on rats. We do the punch biopsy, about 6 millimeter, full thickness skin, and do the study for 14 days. And what we see is the wound close much faster compared to negative control, and also on par with the positive control. And what most important, when we do the histological analysis, the formation of the new skin is good. The structure, we have epidermis, dermis, and no keloid or no excessive formation of collagen is formed. Okay. So next is zingiber zurumbate. Okay, this is called one species of ginger. Okay, and it is long used, we know, as for inflammation and skin ailment, and rich in zerumbone as the active compound and also essential oil. It promotes healing also and regeneration by our research. Okay. Also, there's a lot of in vitro before study have been done. Is not toxic. Okay. And there's a lot of active compounds, especially zerumbone inside there. So they do the topical application, and it is on par, and it also helps in wound healing. And I think this one is already patent. And also the formation, it is found that it also reduces inflammation, especially at the first day. And we know wound healing in 4 phases. They have inflammation phase, they have reconstruction phase, and everything. So basically, what they do is it reduces down the number of protein probably. So probably it contains the bioactive anti-inflammatory compound. Also, it increasing hexosamine and uronic acid. Okay. Very important, especially in tissue remodeling, hydroxyproline in the formation of collagen. Also increase the production of TGF-beta 1, which is very important, which to promote collagen and fibronectin, which is the extracellular matrix production. And also interleukin, it induces the cell to produce interleukin 10, which is very important in reducing the inflammatory response. And the tissue produced is comparable and good compared to positive control. So the last compound is zerumbone. We call it as a bioactive hero. Okay, and it's found in zingiber zerumbate. Okay, that is the crude extract. Now we go to the active compound. Then it's known it have anti-inflammatory, antioxidant, and antimicrobial, and it promotes collagen synthesis and also cell proliferation. So here they do also the in vivo analysis. What they found is what we found is the wound heals, okay, as comparable as using the control with a Socosyl gel. Socosyl gel is the commercially available in the market with 10%, and also okay, and much better compared to the negative control, of course. And the wound contraction can be seen here. It's not very imminent. And also, a lot of protein is produced here, especially collagen, hydroxyproline, and interleukin 10 and everything. Much same with the results from the crude extract. And also the formation of the new tissue is quite good. And they, after 5 time, we call it, after day number 10, you can see the formation of epidermis, okay, especially in the mice that is wound that is treated with the compound of zerumbone. So this proves that zerumbone also help in wound healing and have potential to be used as the next wound healing agent. Okay, so from the side of the lab bench to clinical side, so the process is still long. Okay, when we have a natural product, okay, so there must be a series of research must be done, especially first in vitro, then go to in vivo, and then develop the formulation. At this stage, if you want to save your product, it's better to patent the formulation. Okay, and then go for clinical testing. But clinical testing will cost a lot of money, of course, and can be done only with precipitation of the people that are interested. Okay, probably from the business side. Okay. So current status for our research, particularly preclinical and early formulation stage. Okay, so still in early, and to further enhance the study, okay, we need to do the robust trial and also go for the standardization of the extract. Hi, excuse. Next. Back. They jump. Back. Another one. Okay. So comparative strengths of these 3 plants. Okay, alocasia denudata angler, anti-inflammatory and wound healing. And Lucy indica, it have anti-inflammatory properties, antioxidant, wound closure, and zerumbate rich in zerumbone, strong bioactivity, especially in inducing wound healing. So the challenges and opportunities, especially in developing product wound healing, especially is regulatory approval authority. And in Malaysia, natural product categorized based on the composition of the intended use. It can be as a traditional use. The traditional use, a traditional medical, we call it medical system. We have Malay traditional medicine, Chinese traditional medicine, Indian traditional medicine product. Okay, and also can be finished herbal product. Herbal preparations ready to consume. Okay, this is sold in market. However, must be get approval, especially from certain bodies of approval. Also can be categorized as homeopathic medicine for prepared according to the homeopathic principle, and also natural product with therapeutic claims. This product claims specific health benefit claim, so this require additional evidence for efficacy. Sorry. Okay. Another tak nampak. Also, the challenge is to standardize the extract. Okay, to standardize because plant have varieties. Okay, are you sure that the plant is right? So, what about the content percentage of it? Okay, must be standardized. That is one challenge. Okay, and also opportunity, but it gives opportunity. Okay. Eco-friendly integrative wound therapies. So because natural product itself is rich source for bioactive compound, also it have the multi-functional activity, and of course it can bring cost effectiveness, especially in the process of treatment of wound. So for regulatory approval, especially in Malaysia, okay, it is controlled under NPRA, so National Pharmaceutical Regulatory Agency, and under Drug Control Authority, DCA. And also, to promote your product, you must have the iklan, the commercial, okay, must have the authority, we call it kelulusan atau daripada Ministry of Health. If not, it is considered wrong and your product can be confiscated or you may be fined, the company may be fined. Okay. In conclusion, the plant that we research, all of the 3 plants and Zerumbun have potential to develop as anti-cancer agent. And translational research and clinical are the only way to know the benefit and effectiveness of this. For our research, mainly now, we are going towards the we want to know the interaction of the component of the active compound with the tissue itself. So actually, what we do is, sorry, So now, not we looking into the molecular aspect of it. Okay, for E. indica alocasia, now we go for the rabbit excision model. Okay, and see the expression of genes that involve in associated in healing, inflammation, and tissue generation. We are using quantitative RT-PCR and also RPPA. Then determination of the metabolomic now profiling by LCMS. And also, when we found the compound which interacts, you can predict the interactions between the chemical compounds in these plants and the specific target proteins by using molecular docking research. So, in silico for the next research. Okay, and for our research, nature source is the best alternative way to find the alternative for wound healing. Okay. So I would like to thank to our research team, especially in CORE, in FSK, UKM, and we business grants, and we do our research at CORE, Biomedical Science Program, and I invite the collaboration from the UE side, okay, to increase the benefit of this research, especially in not only natural wound healing, also in the natural product development and so on. With that, I thank you very much for listening, and thank you.
Yeah. Thank you very much for a nice presentation. Also interesting presentation, Dr. Mazlizam. So I would like to a little bit summarize the presentation. So there are at least 3 natural products or plants that can wound healing treatment, treat the wound healing, and then now the current status from this development of the product for wound healing in preclinical and standardization, yeah. Okay, and then there is also challenges for development of drug or wound healing treatment. Is about the regulatory and of our government, maybe, yeah, our ministry. So okay, I think we can make collaboration to develop this because we are in the region, yeah, the same region, and we have so many natural products in our country. So I think our PhD student has also research in this wound healing, yeah. She is dermatologist and she's now finished in the finishing of the dissertation, and I think later we can make a collaboration in this development of natural product for the wound healing. Yeah. Okay, thank you very much for the two presenters. Now we will open the discussion. So please, please come forward, Professor Novi and Dr. Mazlizam. We discuss more deeply maybe for this presentation. So I invite you, all of the participants, to ask or to give the question. So Dr. Radiana from our department, from our faculty, she come from the histology department. So is there, Sadam, ada ini, so please, ininya apa, mic-nya. So, nggak ada yang ini ya. Okay, thank you, Dr. Radiana. Please.
Thank you so much for the inspiring lecture, Prof. Novi and Dr. Mazli Alam. So let me ask the guests first. So, we are working with different settings. So we are coming from stem cell research, but the way we are doing it now is that we are trying to emulate stem cells as drugs. So all the steps that you discussed previously, in vitro study, in vivo studies, and really kind of like making a preparation before implanting to patient for clinical trials, that's the way that, or the route that we will take. Now, regarding your models, I'm interested to know, because this is what we see in the stem cell implantation, whether we place it in hydrogel, collagen, or in other type of microencapsulation of the stem cells. When we do an animal model with multiple burn injuries, there is a chance that paracrine effect of the stem cells synergize to the other wound area, and thus causing it for us very difficult to have statistical significance. So it's usually when you place the stem cells in one of the wound area, but the other wound area is not supposed to be having any effect, they do show some improvement or repair. So do you also see this kind of phenomena in your models? That's number one. And number two, it's about, generating standardized burn rat model for the injury itself and to really show the efficacy. So that's two. For Prof. Novi, Prof. Novi, can I ask about real life application? If we are already considered obese, what type of diet or maybe fasting is appropriate for us to reduce our condition and keep us healthy? Thank you so much.
Okay. Thank you. Thank you, doctor, for the question. When we do the implantation, in my previous experience, before this, I'm doing tissue engineering. So it's normal to mix stem cell with biomaterials and put it inside the animals, okay, to know whether your construct or whatever your things work or not. Okay, so you must consider also this synergistic effect, okay, or we call it as cakap Malaysia lah. dia mesti ada effect. When you implant something inside the animal, okay, the thing will get inside. So there must be a chance of this certain growth factor or these certain chemicals we go to the body and then can interfere with any of your wound. So what we do, you usually, my previous presentation, if you have one stem cell with one construct, you just implant it into one mice and do not mix your wound. You put this compound A and the next one you put compound B, and then you expect the result will be different. No, you must consider there will be the active compound can go inside the body and can produce certain precursor, certain secondary metabolite which can affect the other place of the wound. So the design of the experiment, if you want a very certain result, you might isolate the mice. Use the same population of mice, however, isolate whatever you want to test. So that's my opinion. So my previous work, we do like that, implant. Okay, we have stem cell, we have muscle cell, whatever is adipose tissue and everything. We separate them, and maybe we do induce it with some drug for them to become something, to produce something. Please try to isolate, not in the same mice setting. The second one is about what?
Second one about the standardized burn injury.
I've never do the burn injury. However, I do the open wound. So usually we use, what we use here is punch biopsy, about 6 millimeter. So we standardize all the area of the wound, just 6 millimeter. And the deep, usually we go for full thickness wound. That means until the fascia. Okay, to really make sure that we want to see the formation back of the dermis and the coverage of epidermis. In big animal model, usually you can also cut using 3 millimeter, 2 millimeter, okay, and maybe you can put satu halangan di keliling luka tu supaya the area also, that's for tissue engineering purpose lah. But
yes,
for burn, I'm not very sure, but we do like that.
Okay. I mean, looking at your picture about the rats, it seems like you have a multiple punch biopsies on the back of the rats. So that's why I'm asking you.
But there's only one compound. It's not
one compound.
Yeah, only one compound.
Okay.
Not like A,
one
B, C. No,
do not mix. One group only receive one compound.
Just one only.
Okay, so that's how you able to obtain the statistical significance in the end. Okay, thank you so much.
Okay, thank you for the question, Dr. Radiana. Of course, for the diets, balanced diet is the best for every person to prevent obesity. According to our research, in 50 obese male, we divided into 25 subject control and 25 subject intermittent fasting. Both of them give the balanced diets, and we see that the MDA, malondialdehyde, as the oxidative stress markers, reduce in both of group. But in the intermittent fasting group, the decrease of MDA is higher compared to the control group. And the other methods to prevent obesity, of course, physical activity, but it is really challenges, as I mentioned before. And the other technique is calorie restriction. The other form of calorie restriction is intermittent fasting. Yeah, intermittent fasting. Many research already proved that the intermittent fasting can stimulate FOXO3 transcription factors, which regulates many genes. One of the genes is antioxidant gene activation. So the activation of FOXO3 can increase the antioxidant to prevent oxidative damage. And our research in 50 obese male also proved that the intermittent fasting also can reduce body weight for 8 weeks. Is it answered?
Would you also recommend like mTOR inhibitors or sirtuin-1 based supplements to, for healthy
Yeah.
aging?
Yeah, sirtuin-1 is really good, yeah, because sirtuin-1 is the acetylase and regulate the mitochondrial biogenesis, yeah. Of course, it still need further research, yeah, to recommend whether we have to take sirtuin. I don't know whether currently already available the sirtuin-1 drug or preparation, but for the mTOR inhibition, there is the natural compounds which can inhibit mTOR. If I'm not wrong, it's reverse-stator, yeah, reverse-stators. It is related with the healthy aging and can extend lifespans.
Thank you, Prof.
Thank you very much. Is there any question? Yeah, from the UKM, Professor Nur Fadila.
Thank you for a very nice presentation, Dr. Novi and Dr. Mazlizam. Okay, my question is to Dr. Novi regarding the FOXO3. Okay, it is a good biomarkers for early senescence or for diagnostic, or is it just dedicated for stress-related senescence? Number two is that sirtuin is also has been implicated in AD. So how we want to differentiate between sirtuin that will lead to AD, Alzheimerism, and also sirtuin that will relate to senescence. Thank you.
Okay, thank you for the question. Actually, FOXO3 is the not the biomarker for cellular senescence, because the biomarker of cellular senescence, the gold standard, which is mentioned in the journal, is S-abigal senescence associated beta-galactosidase. This is the enzymes in the lysosome, and the activity this enzyme is increased when cell undergoing to senescence. However, the FOXO3 is a transcription factors which is regulates many kind genes. One of the genes that regulates by FOXO3 is antioxidant such as SOD catalase, yeah. Various research also prove that the increase of FOXO3 associated with the healthy aging, because it can extend lifespan, but the result is still limited in the animals models. Yeah, so for the gold standard of biomarkers until now is S-abigal, and also the proteins such as P16 and P21. This is the inhibitor of protein kinase, cyclin-dependent kinase. Yeah, however, the FOXO3 usually also measures to detect the mechanism how the cell protects from the oxidative stress, because oxidative stress is related to the cellular senescence. It's the cause of cellular senescence. And the second question about this sirtuin. Sirtuin-1 is the acetylase, yeah, which regulate also FOXO3. If the sirtuin-1 increase, the expression of FOXO3 also increase. And Sama disease? What is?
Alzheimer.
Oh, Alzheimer disease.
Yes.
How to differentiate?
Yeah. Sirtuin-1 also involved in progression to Alzheimerism.
Oh, the expression, the high expression of
Yeah,
sirtuin-1.
sirtuin-1, yes.
Oh,
Because our group study on Alzheimer, and yours is actually on senescence.
so the expression of sirtuin-1 is increased in Alzheimer.
It will lead to progression of
The
Alzheimer's.
progression.
Yeah.
Yeah. Oh,
So how to differentiate whether the sirtuin is actually for Alzheimer or for senescence?
yeah, it's
Because they are two
difficult
different.
to answer, I think, because in other hand, sirtuin-1 is the have the beneficial effects because it is can activate the mitochondrial biogenesis. Yeah, maybe maybe in Alzheimer's different cases, we have to explore further by more deeply research. Yeah, why it's increased. Yeah, if I want to ask, the increase is mRNA level or protein level in the Alzheimer's? Protein levels in the patient of Alzheimer or animal models? In the patient. The patient from the blood. Because the sirtuin, the extent of sirtuin in the within the cell, yeah, not in the blood. So maybe if we can detect sirtuin in the blood, it means that there is the lysis cell, maybe, yeah, lysis cells. So the sirtuin release because the action of sirtuin within the nuclei, yeah, to activate coactivator PGC1s for mitochondrial biogenesis. Maybe this is a response, maybe I don't know the exact answer, I'm sorry.
Okay, thank you. Is there any question? One question more? Because our time is going up. Is there a question? No? I have a question about to Dr. Mazlizam. You have already presented about the compound or content of the plant that may treat the wound healing, yeah? The wound, yeah. So the potassium, it can be that all of the plant, that because we are rich in natural product, yeah, in Indonesia and also Malaysia, it can be that the plant that has potassium in the content of in this plant, yeah, it can be used to this wound healing. What do you think about that? The potassium, because the potassium is so high in your research with the plant for the wound healing.
For this investigation, yes, we found it's quite high. However, there is a certain cascade, especially in sodium palm and everything, that probably have the effect of that. So what we found is actually elemental research. So probably we have to investigate more on that findings. But we can always try, if we have a potential plant with potassium itself, probably we can just try and do some studies on the ability, whether we can improve the healing process itself. Yeah, but probably not all potassium. Probably some can cause toxic. We do not know. Okay.
Yeah, we should investigate
further. We should investigate
this mechanism also. Okay, thank
together.
You. Okay. Yeah,
thank you.
We can make the research collaboration. To Professor Novi, I have also curious about this cellular senescence mechanism. Yeah, I read that the cellular senescence also has benefit and detrimental effect, yeah, such as actually benefit also for especially in cancer, because this is the state of the cell stop but remains viable. So it means that if we treat with one of the strategy, it can be also treat the cancer, or it can be also induce the proliferation, or what do you think about both sides of the effect of this?
Yeah, thank you, Prof. Asmarina, regarding the question. Yeah, right. Yeah, the cellular senescence have the beneficial effects. Yeah, because cells stop to divide, especially in cancers. Yeah. However, currently in the cancer, cellular senescence also have the bad effect. Yeah, because the cellular senescence is metabolically active, can secrete SASP, senescent associated phenotypes, which consists of cytokine and chemokine, and it can stimulate the cancer progression also. And according to the research, that cellular senescence have the benefit in the young organism, yeah, for the wound healing and for the other mechanism. But in the old organism, cellular senescence have the more detrimental effects here, because SASP can exert harmful effect, can influence neighboring cells to senescent again, yeah, and also it can stimulate tumorigenesis also, yeah, if within the cell there is the premalignant phenotype in the cell.
Okay, thank you very much. Yeah, thank you very much again for the presenter for nice and interesting topic. Yeah. So yeah, now we are in the apa ya coffee break ya, coffee break session. There is no coffee break session. Okay. So yeah, okay, we will give a present to the speaker. Yeah.
Thank you very much, Professor Asmarina, Professor Novi, Dr. Mazlizam, and also the participants. We would like to invite Professor Dr. Wawaimuli Arozal and Biomed PharmD to come to the stage to give a goodie bag as a token of appreciation for the speakers. And please stay for the photo session. Professor Asmarina, Professor Novi, Dr. Mazlizam, come to the stage. Thank you.
We will now proceed with the rapid fire session 3, where selected postgraduate students will deliver presentation of their research. Kindly make your way to room 1 or room 2 to show your support. To the participants, this room is as listed in the program booklet. If you have any questions, our committee members will be available to assist you.
We would also like to inform everyone that we will gather again at 11 for the closing ceremony. Winner announcement and award giving will also be held during this closing ceremony. This must be the moment that we're all waiting for, especially for those who participate in the competitions.
ladies and gentlemen. Okay, welcome to Rapid Fire Session 3, featuring the topic of molecular biology and proteomics health community. Okay, before we begin, allow me to introduce myself. My name is Aulia Fitri, and today I will be serving as the timekeeper. Okay, now I will introduce to you our judges for today. Okay, first, we have Mrs. Arum Naruni, SSE DEA PhD,